Hi everyone, I'm Nadia Selami, PacBio Segment Marketing Lead for Biopharma Applications, and I'll be the host and moderator of today's webinar. I want to welcome everyone to this webinar with today's topic focused on the use of HiFi sequencing for antibody development. I would like to introduce our speaker and expert on antibody development, Dr. Fortunato Ferrara, the VP of Discovery at Specifica and IQVIA Business. In the first part of our webinar today, we'll hear a 10-minute intro by me with an overview of HiFi sequencing, how it compares to other sequencing technologies, and its application in the antibody discovery and development process. This will then lead us into the talk by Dr. Ferrara on how he uses HiFi sequencing in his antibody development work. And we have a lot of great material to cover today. Both the introduction and Dr. Ferrara's presentation are pre-recorded, but will be followed by a live Q&A session. You're welcome to submit your questions at any point during the webinar by typing them in the area provided on your attendee control panel, and we'll answer as many questions as we can at the end. We've also uploaded complementary literature to your control panel, so please feel free to download this and take it with you. We'll be recording this webinar and making it available for download in the coming weeks. So please keep an eye out for a follow-up email with a link to the recording. And immediately following this webinar, you'll receive a brief questionnaire. Please do take the time to fill it out as it helps us understand your needs and plan for future webinars. You can stay up to date on upcoming webinars and events by visiting and bookmarking pacb.com/events. So with that, let's get started with the talks before we have the live Q&A. So let's talk about HiFi for antibody development and a quick intro to HiFi sequencing. PacBio provides highly accurate HiFi sequencing with which you can have confidence in your results. We provide powerful research tools for target identification and biomarker discovery, mRNA vaccine sequencing, directed evolution and phage display as used in antibody development, AAV gene therapy and gene editing, as well as plasmid sequencing. Using these tools can enhance your discovery process, making your development more efficient and will ultimately enable you to be more confident in your results and decisions. Let's talk about what HiFi sequencing is. Given their central importance to PacBio long-read sequencing methods, let's take a closer look at how we generate HiFi reads using smart sequencing. As illustrated here, HiFi reads are produced using the circular consensus sequencing (CCS) mode on PacBio long-read systems. In this sequencing mode, the polymerase makes multiple passes around the same DNA template to generate an intramolecular consensus read for that single molecule. This process results in a HiFi read that is both long, up to 25 kilobases or so, and can achieve a very high median accuracy of 99.9% or greater. HiFi reads have many benefits, such as long read lengths up to 25 kb, high read accuracy of more than 99.9%, easy library preparation, low coverage requirements, small file sizes to minimize compute time, and a single technology solution for a range of applications. As a result, HiFi reads produced through the CCS sequencing process are the most common data type being generated with PacBio sequencing today. And as you can see in the top right corner, HiFi reads combine the accuracy and read length of other technologies in one. Another unique feature of HiFi sequencing is that it can also deliver epigenomic data through the simultaneous detection of DNA base modifications during the sequencing run by analyzing the polymerase kinetics. The way this works is that the DNA polymerization rate is slowed whenever the polymerase encounters a modified base in the template strand. For example, think of the bulky methyl group that's found on the modified A base highlighted here as acting like a molecular speed bump. By normalizing the slowed incorporation rate against an in silico kinetic model for an unmodified DNA template, The resulting spikes in this type of interpulse duration ratio plot, shown on the top right, can be used to infer the presence of modified bases in the template. This type of kinetic information can be automatically generated and processed during a HiFi sequencing run and does not require any additional complicated biochemical sample preparation methods. With an optimized polymerase and no amplification or cluster generation, HiFi sequencing provides uniform coverage across sequence contexts, even for high GC regions and tandem repeats. HiFi can readily sequence through regions that other technologies struggle with. These are AT-rich or GC-rich regions, highly repetitive sequences, long homopolymers, like for example mRNA poly-A tails, and palindromic sequences. Okay, let's now discuss some of the key performance characteristics of HiFi data that's generated through smart technology. Several key strengths of HiFi data include: first, obviously, are long reads. We have the ability to generate long reads that span tens of kilobases in length for sample library insert sizes, ranging from as short as a few hundred base pairs to over 20 kb. With regards to accuracy, because of PacBio's non-systematic error profile, HiFi sequencing data can achieve Q50 or 99.999% consensus accuracy. Furthermore, HiFi sequencing is unique in that it provides single molecule long reads with a high median accuracy of more than 99.9%. And because of the lack of any requirement for PCR amplification during sequencing, with PacBio you can sequence uniformly through varying levels of GC content and genome complexity. And finally, with smart sequencing, there is no need to perform any separate sample preparation steps to capture epigenetic information with your sequencing data. Okay, so that's it on HiFi sequencing and its benefits, but now let's have a look at how HiFi sequencing compares to other sequencing technologies. The Human Genome Project published the first human genome, but many parts of the genome were still missing from this. The first complete human genome, spanning from telomere to telomere, was only recently completed and could only be built from HiFi reads, as they provide both the accuracy and read length to cover the previously inaccessible regions of the genome. When you look at how much of the full human genome sequence different sequencing technologies cannot access today, you see that it ranges from 28 megabases to 200 megabases, with PacBio HiFi reads missing the smallest fraction of the human genome, while common short-read technology misses the equivalent of chromosome 3 due to its technological limitations. Not only are HiFi reads able to cover more of the human genome than short-read methods, it also matches them in accuracy while providing up to 100 times longer reads, as was demonstrated in the Precision FDA Truth Challenge. The accuracy and read length that PacBio HiFi sequencing unites leads to this technology having the fewest variant calling errors when compared with other common sequencing technologies, spanning from single nucleotide variants to indels and structural variants. All this makes HiFi sequencing a great tool to enable your research. And we're constantly working on making our chemistry even better, more affordable, and provide instruments at a throughput that suits your applications. So let's look at what is coming in 2026. We have two instruments on the market, the benchtop Vega instrument and the higher throughput Verbio. A larger high-throughput instrument is currently in development. The Vega is our benchtop instrument that generates HiFi reads, including 5-methyl-C detection when sequencing genomic DNA. Coming in 2026 is the additional detection of 5-hydroxymethyl-C, new Spark sequencing chemistry that allows for 4 times lower DNA input needs, and the ability to run sequencing within a single shift by enabling shorter movie times, such as, for example, 6 hours or less for inserts between 500 and 2000 base pairs, as you would likely use for your antibody development. Also, coming in 2026, Vega will enable compliance with 21 CFR Part 11.
For Revio, we also have exciting new features coming next year. As Vega, Revio will be able to also provide 5-hydroxymethyl-C information on every run on genomic DNA. It will have stronger compliance support with detailed logging of instrument actions. But most excitingly, we're introducing multi-use smart cells and new sequencing chemistry that can drive the cost of sequencing down significantly. And last but not least, we already expanded the use of Kinnex kits to amplicons. Kinnex improves throughput for smaller fragments by concatenating cDNA or genomic DNA amplicons to take advantage of the full read length that HiFi sequencing is capable of. With the Kinnex kits, you can increase throughput for amplicon sequencing between 8 and 12-fold, depending on the amplicon length. This is ideal for many biopharma applications that typically use shorter fragments like scFv and Fab antibody discovery, mRNA vaccine characterization, and gene editing assessment. And speaking about antibodies, this is the topic of today's webinar, and I want to give a brief introduction how HiFi sequencing can be used in combination with phage display approaches to enhance the development of biologics such as antibodies. So here's an overview of how HiFi sequencing fits into the phage display antibody development process. After libraries are generated synthetically or by immunization and panning is performed, amplicons of the antibodies are sequenced on a PacBio instrument. After clone selection, the resulting proteins can be produced and biochemically and biophysically characterized to identify the ones that match your selection criteria. HiFi sequencing enables your ability to identify novel antibodies with desired characteristics such as certain antigen binding affinity and kinetics. This is enabled through several characteristics of HiFi sequencing. You can characterize full-length plasmid or amplicon libraries up to several kb. Through the combination of read length and accuracy, you can analyze multiple mutations or variants per molecule. Difficult to sequence regions, such as repetitive or high GC content areas, can be accurately resolved. And the read depth that HiFi provides allows you to increase throughput compared to traditional Sanger sequencing approaches. And when we look at antibody development in more detail, HiFi helps overcome limitations of other technologies. For example, the scFv molecule is typically around 850 base pairs long. Short-read sequencing cannot cover the full length of this or other important fragment types that are used in antibody development, which is why typically Sanger sequencing is used. However, Sanger sequencing suffers from low throughput and high hands-on time, and thus limits the number of clones that can be analyzed. HiFi can cover the whole molecule at Sanger level accuracy and greatly increases the number of clones that can be analyzed, which increases your chances to find the ones that match your desired characteristics. With this, I would like to hand it over to our speaker for today, Dr. Fortunato Ferrara, who will tell you more about how he uses HiFi sequencing in his antibody development work.
Hello everyone, my name is Fortunato Ferrara, and today I'm gonna talk to you about our application of PacBio for our in vitro discovery. So why we decided to use PacBio over other NGS platform like Illumina? So most of our selection campaign are conducted using single chain. A single chain is a construct where we recapitulate the binding specificity of an antibody, but it's only composed by a variable light chain joined by a linker to a variable heavy chain of an antibody. Of course, after a discovery campaign, people are interested in reformatting these single chain, which are very good to be used in in vitro platform like Phage and his display. They want to reformat them as full-length IgG for diagnostic or therapeutic goals and purposes. In order to do that, it's very important to have the pair information of the VL, the variable light chain, and the corresponding heavy chain of that particular antibody. The issue with an NGS platform like Illumina, while the depth is very deep, So we can achieve 10 to 250 million reads. That's usually limited for only one of the variable chain. For instance, the heavy chain, which by definition is the main driver of the antibody specificity. So with Illumina, we can have usually around 300 base pair. So we can go in both directions of the variable domain. And in this way, by overlapping the sequence result, we can have a lot of sequencing, but only focus on one domain, completely missing the other pair domain of the single chain. So there have been some publication in which there are some strategies to try to pair, but using Illumina platform, the light chain with the corresponding heavy chain, but those are cumbersome and they are not straightforward. So that's why we decided to apply PacBio when at the end of our discovery campaign to have the full information of the pair light chain and heavy chain. So PacBio compared to Illumina platform is a less depth, usually between 2 to up to 10 million. They are coming up with a new platform, the Kinnex, and we are going to be beta test this kind of platform. And the promise is that they are going to be able to go deeper, probably close to 100 million reads. So this is the typical antibody drug discovery journey that we conduct here at Specifica. So we start from the library construction, where the QC is performed via NGS and Sanger sequencing. There is the actual discovery part of our process, where this library is challenged against a target of interest. So we are looking for antibody in our library that are specific for a particular target. So then we can perform antibody optimization. The data that we collect during the discovery campaign can feed AI model for their validation. And then Specifica is part of the Acuvia family, where the lead antibody identified during a discovery campaign can be further validated in preclinical study, but all the way into clinical trial, thanks to all the different laboratories that belong to Acuvia. For today, I'm going to just focusing about our use of sequencing in general and PacBio sequencing in particular during our discovery campaigns. So this is an overview of our discovery platform, discovery strategy. We built a very large library using phage display. So in this way, we can have over 10 to the 12 phage particle displaying a unique antibody that can be tested and challenged and enriched for a specific target of interest. So we usually perform 2 rounds of phage selection. So that allows us not only to enrich our antibody population, but also to obtain a size, a number of clones, that it's very easy to transfer into any display system. The advantage of this display is that we can use flow cytometry, so we can have a real-time assessment of how our selection is going. And so that allows us to really fine-tune the property of our antibodies in terms of affinities, specificity, and functionality. So like we can specifically select antibodies that, for instance, compete with a ligand, so they can be agonist or antagonist. Usually at the end of a discovery campaign, we either rely on random clone picking and Sanger sequencing. That's usually when we are looking up to 100 clones, but we can expand, and I'm going to talk about deeper about that, our antibody portfolio against a specific target via NGS and usually via PacBio sequencing. So let me talk about how we routinely apply sequencing in general in our pipeline. So Specifica does early stage discovery. So we apply definitely sequencing during the library construction to quality control our libraries, during the step of the selection to understand how our selection is going, and definitely at the end of the selection campaign to identify the lead candidates. So all these steps are subject to sequencing, either Sanger or NGS, and all the data that we collect and the further data after the validation in terms of developability, binding, and kinetics of the antibody can feed back our bioinformatics tools, so we can start having some idea about sequencing and what kind of properties a particular sequence provide to an antibody. So let me exemplify some use case studies. So for instance, library QC and validation. So we try to be very transparent in the way we build our antibody libraries, because the quality of our library is the key ingredient to have a successful antibodies discovery campaign. So, and like I mentioned, when we build a library, we deeply QC the library via NGS and also Sanger sequencing. And we were some of the first people to really go deep into the quality control of our libraries. And this is, of course, possible because of NGS. So we build what we call a generation 2 library. Those are usually human-naive libraries that use all the natural germline. And the first step in terms of QC of the library, we use MySeq to really appreciate the diversity of the HCDR3 of this library. The HCDR3 is the main driver of the specificity of an antibody. And that's basically, the more diverse is the HCDR3, the larger is your library and the more potential specific binder for different targets you have in your library. So not only we do MySeq at the HCDR3 level, but we also use PacBio to, of course, given the limitation of PacBio in terms of 10 million, our library are almost 100 million to almost a billion different HCDR3. So PacBio give us only a snapshot of the functionality of the pair VL and VH, but still being able to analyze 10 million pair sequence, it's a good QC step in order to really appreciate the functionality of antibodies. What I mean with functionality is that basically the library is properly built, that there is good pairing of VL and VH, there are no frame shifts, stop codon, and of course that's possible only with PacBio and not with Sanger sequencing, because Sanger sequencing is way limited in terms of number of clones that you can screen. We still use Sanger sequencing most of the time during the validation step. So what I mean with validation is like we test our libraries against some target, established targets, so benchmark antigens if you want. And at the end of those kind of selection campaign, we usually rely on Sanger to identify between 100 and 200 clones to really validate the efficiency of our library in terms of selection. We also have generated what we call generation-free libraries in different format, single chain, Fab, but also single domain or VHH library. And generation-free are what we call semi-synthetic human libraries. So the framework of the antibody, the scaffolds, if you want, are based on therapeutic antibodies, which have already been approved for clinical uses. And what we do is like we, in this library, have all the CDR but HCDR3, which are derived from one of our large data set of antibodies. Those CDRs are purged of sequence liabilities. What are sequence liabilities? Are amino acid sequences that might become problematic when we further develop an antibody for therapeutic uses. So they might affect the antibody stability, the antibody production, and whatnot. So the CDR, all the CDR, but CDR3, HCDR3, the CDR3 in the heavy chain are purged of that and are used for our library construction. The HCDR3, the diversity, natural diversity is really large. So it's hard to synthesize right now with the technology in place, HCDR3 libraries, if you want. So we extract that diversity from almost 1 billion cells obtained from 10 healthy human donors. And by doing so for each of our library, we also have an exclusive library, because all those 10 healthy donors are always different for any library that we prepare, either for internal use or for our customers. And once more, all those libraries, regardless of the format, were deeply QC'd using NGS, similar to generation 2 library, MySeq to really appreciate the diversity of the ACDR3, PacBio to really appreciate the functionality of the clone library, and Sanger during library validation. But we also use sequencing in general during our discovery effort. So the first publication on the top, it was just a discovery campaign where we did to rapidly identify antibody against SARS-CoV-2 RBD with neutralizing properties. And on that particular case, we rely only on random picking and Sanger sequencing. The next step is we also do affinity maturation of our libraries, of our antibodies. And for instance, in that particular paper, we describe our affinity maturation strategy of a therapeutic antibody against B7H4. And in that particular case, we use MySeq in the design phase, and we use Sanger during the validation. But if we want to expand hits, so if we want to expand the portfolio of antibody against a given target, in this particular case, the RBD from SARS-CoV-2, that's when we really start using PacBio together with Sanger sequencing. So PacBio, once more, give us pair information, so VL pair with the corresponding VH, and we can go deeper into the analysis of our selection output of a population of antibody extremely specific for a target of choice. And also we have been using PacBio to generate what we call NGS data packages, which I'm going to describe later in this presentation. So let's go deeper into, if you want, rank order prioritization and hit extension. So what we found out when we were comparing Sanger result with NGS obtained via PacBio is that what you can see here is that, let me just get my laser pointer. So the clones in orange obtained after Sanger are good enough to recognize the most abundant clones. So as you can see here, and so in gray are the clones, the most abundant clones identified by using Sanger sequencing. What NGS does is not only confirms the top ranking clones, but for instance here, after the top 7th clone identified via Sanger, between the 7th and the 8th most abundant clones of Sanger obtained after Sanger sequencing, we see that there is this gap. So not only we improve the order, the rank order, but also we can fill the gaps, expanding the hits identified for a particular target. Not only that, so if you can see here, so here I think there are about the top 11 clones identified via Sanger, and those 11 clones, they represent basically 97% of all the clones identified by NGS sequence in terms of abundance. So there are, in that 3%, there are all these clones that you can see here. So they are low abundance clones, which you are only able to identify via NGS because it will require a massive screening if you rely on Sanger. And what we did was not only we express the antibody obtained with Sanger and the most abundant antibody identified via NGS, but we also express a lot of clones with very little abundance, a lot of clones that were in the 3%, in the missing 3%, if you want. And we found out that over 70% of those clones are binders. So not necessarily they have strong affinity, but they were binders. So relying on NGS really help us to expand the antibody diversity at the end of a selection campaign. What we learned during this attempt to expand the portfolio of antibody against RBD. So what you can see here, those 3 red triangles are free antibody that we use as benchmark at the very beginning of the SARS-CoV-2 pandemic. This pink circle here are the antibodies identified just by random picking and Sanger sequencing. As you can see, the affinities, this is an isoplot affinity plot, iso-affinity plot. As you can see, even just relying on Sanger, we can identify a very high affinity binder here. What happens when we start expressing all these clones identified by PacBio? As you can see, some of the clones, they have worse affinity than the one identified by Sanger. A lot of them they fit in the range of affinity that the clone identified by Sanger, a range of affinity similar to the clones that we identified by Sanger, but there are also a remarkable number of clones with even better affinity. So in our experience, it's really worth, if you really are looking for covering the larger diversity of possible binders for your target, to not only rely on Sanger, but expanding your antibody diversity using NGS, in particular PacBio. So then we analyze, okay, does abundance relate, for instance, with affinity? So as you can see here, basically what we did, we sequenced via PacBio 2 steps during our selections campaign. So 1 nanomolar was the final antigen concentration. Is a very relatively low concentration of antigen that we use for really push a population in terms of affinity. But also we sequence 1 step before the final sort, if you want, the final enrichment step, by using slightly less affinity, and those 2 populations were sequenced. Of course, there is a very good correlation between those 2 populations, meaning the most abundant clone that you see in the last sort is also the one that you obtain being a bit less stringent in your selection, and there is a very good correlation among the clones. But what happens when we try to correlate frequency with affinity? In other words, is the most abundant clone the clone with the highest affinity? And the answer is, at a glance, definitely not. So if that was the case, here you will find a lot of dark blue dots, because the darker the blue, the better the affinity. But as you can see here, there is a discrepancy between frequency and affinity. For instance, the most abundant clone has an affinity between 1 and 10 nanomolar. The second most abundant is between 100 picomolar and 1 nanomolar. So already here you can see the discrepancy. This clone, which is relatively high affinity, it's over 1 micromolar. So definitely not a good binder, but it's relatively high in terms of frequency. So here you have a clone which is relatively not abundant, with an affinity below 100 picomolar. So the first lesson learned is you cannot really rely on abundance and affinity. But then we went even deeper. So thanks to NGS, we not only identify antibody belonging to different cluster, but we also identify antibody of several antibody that belong to the same cluster. What do I mean with cluster? So at a glance, cluster, for instance, are antibody that are very similar, if not identical, ACDR3 in terms of amino acid sequences, but also in terms of biophysical properties. So let's say the ACDR3 is the same. The antibody can vary in all the other CDRs, in the framework, but the idea is that since they share the same ACDR3, most likely they have similar function in terms of epitope recognition and in general as the binding properties. So what we found was, okay, so if we explore within a cluster the abundance of the different antibody, what we saw is there is, again, little or no correlation between abundance and affinity, as you can see here in the plot. What I mean is not necessarily the most abundant clone in a cluster is the best binder. And usually when you do Sanger sequencing, that's what you pick up, is when you rely only on Sanger sequencing, you are able to identify the most abundant clone in a cluster. You are losing most of these clones that belong to the same cluster, but that might have very low frequencies in the entire population. but the correlation improved a bit. Like the score is not great, but it's definitely better than before. There is improved correlation when you look among cluster, meaning if you rely only on the most representative clone of a cluster, although there is a chance there is not going to be the best binder in that cluster, there is a correlation between the top clone in a cluster and affinity when you compare the top clone in different clusters. Let's go even deeper into that. So here what you can see is like the clone with similar colors are clones that belong to the same cluster. And what we saw here is that this particular clone and this particular clone, the frequency was 0.02% frequency. This clone here was 0.6% frequency. So there was a 300-fold difference in frequency, but this very rare clone had 100-fold improved affinity compared to the one with a 0.6% frequency. Meaning, if I have to repeat myself, that there is very little correlation where you are able to collect so many data thanks to NGS of between affinity and frequency. But the take-home message is also that 70 to 90% of the time in your cluster, you get clones that have similar or worse binding compared to the most abundant clone in that particular cluster. But sometimes, 10 to 30% of the time, you might get what we say lucky, and you can find out a clone in the same cluster with better affinity compared to the most abundant clone in the same cluster. So, and this is the total representation, the graphic representation of what I'm saying. So as you can see here, in general, and that was possible because we expressed and validated, characterized over 300 clones after NGS analysis. And you can see that one more, 10 to 30% of the time, you have improved affinity, 10 to 30% of the time, you have similar affinity, and 40% of the time, more or less, you have worse affinity, but they are still binders. All this very detailed analysis is possible, of course, only because we went deep into the analysis of our output thanks to PacBio sequencing. So now I move on to our latest use of NGS and PacBio, and it's about providing data for AI training. So everybody is aware that recently AI and machine learning models are improving a lot, and they're doing steady progress in terms of protein structure prediction and also de novo antibody synthesis or de novo of antibody improvement. But they need data. So all these platforms need high quality data. And within the in vitro platform, like phage display libraries and selection use phage and its display is a way to provide those high quality data. In particular, we are very confident that our platform, based on several selections that we have conducted for different clients and also for internal R&D, at the end of a selection campaign, we generate antibody that are highly functional and they have good quality and are good quality antibodies. So our data are valuable for training AI and ML models. We rely on PacBio because PacBio can generate these pair-end antibodies, so VL in pair with VH, and it's optimal for obtain the big picture of a selection at the end of a selection campaign. What we say, and usually for us, it's we can really sequence an entire yeast population that has been enriched for antibody highly specific for a target of interest. And not only that, so to train a model, you not only need good quality positive data, but you also need negative data. And again, what I mean with that is like we can sample some of our library in yeast, and instead of focusing on antibody which positively bind your target, we can also enrich for antibodies that are bona fide negative for that particular target. And that's, again, it's possible to go deep into this analysis thanks to NGS and PacBio. Why I'm stressing out that PacBio is our NGS platform of choice for this? So here, very briefly, this study here showed that even with a smaller data set of pair data, VL and VH, obtained, for instance, with PacBio, the AI model, the training of the AI model is more effective compared to a larger data set, which is based only on a single domain, for instance, only on the VH. And that's basically what they did in this study, in a nutshell, was basically by training the model with pair sequence VL and VH as relatively way smaller data set compared to the data set obtained via Illumina and focus only on one domain of the antibody, the VH, their model was performing much better when we're using pair sequencing in terms of identify, for instance, antibody of healthy individual with antibody obtained from SARS-CoV-2 patient, and they also were able to discriminate better between antibodies generated in patient with flu compared to patient affected by COVID, and even across all these three groups, flu, COVID, and healthy patient, the model trained using pair sequences outperformed the model we use larger data set but only focus on the VH. So what are we offering is these data packages for training AI data, and those data packages are based on PacBio sequencing. So we can train an AI model. So we have in-house, we have already conducted selection against 2 targets, IFN and IL-2, and we have sequenced all by PacBio all the different selection steps, and those data are already available to train AI ML model. We are also working with one of the big players in the AI antibody prediction field, and what they wanted was they identified 10 targets of interest for them, so 10 different antigens, if you want, and we perform a discovery campaign against all the different 10 targets, and we are sequencing all the different steps during the selection campaign, including negative data, if you want, via PacBio, and that's what we are going to provide to the client. And that can be done for any company that is interested in this kind of approach, and it will, so compared to the data package which are immediately available, it might take us 1 to 4 months to generate the data, but we can basically challenge our library for any target of choice. But we can also test your AI model. So we have been approached by companies that they want to design their own library, their own antibody library, and we can generate the library. We can challenge their customized library against up to 6 different targets of choice, different 6 antigen, so they can fine-tune and train the AI ML model. And again, that's require us 1 and 4 months, and all the data are generated by sequencing with PacBio. So at the end of either the training or the testing of an AI model, what we provide to the client is a raw FASTQ file, and we can, of course, sequence different sort concentration. We can strategize with our potential client what kind of question are asked in terms of positive and negative binding, but even in terms of what kind of properties they want this antibody to have, not just affinity, but for instance, competition and whatnot. We can do a preliminary NGS analysis in terms of abundance and enrichment. And again, we can fine-tune all the aspects of the antibody that they think they need in order to train the AI ML models. So why do we think there is an advantage in using our technology in combination with PacBio sequencing? So what we know is, like, we know that if our library is challenged with a relatively high concentration of an antibody, which is 100 nanomolar, can provide, and this is based on several sequencing that we have done using PacBio, between 100 to 10,000 unique ACDR3 for population. Based on some study, including the one about SARS-CoV-2 that I mentioned before, I described before, we know that the antibodies, even when we don't push for affinity, are highly functional. So we have 70 to 90% hit rate. So you can be very confident about the data that we provide you are linked to functional antibodies. But we can also play with different stringency. So we can include population with different degree of stringency, meaning stronger affinity, specificity, and again, we can fine tune for competitor, non-competitor for a ligand. For instance, if your target is a receptor and whatnot, and we can sequence every single step of our selection strategy via PacBio. So we have a case study about the use of PacBio to generate data packages. So we launched what we call the AI competition for antibody benchmarking. So the challenge was we invited all different kind of institutions, from academia to pharma to AI-based company, to answer these three challenges. So all the data that we generate for this challenge were obtained using PacBio. The challenge include to affinity optimization and maturation, prediction from sequencing of an display output, and out-of-library design. I'm going to go describe a bit better those challenges. So challenging one was the in silico antibody affinity maturation. What we did was we identified a SARS-CoV-2 RBD antibody during our selection effort with a relatively high affinity, meaning it was not a super tight antibody against the antigen. What we did, like during the affinity maturation that we conducted for this particular antibody, we sequenced all the mutants obtained during the affinity maturation effort via PacBio. So we provided the parental antibody, which is with its affinity, and all the data set of all the mutants that we obtained during the affinity maturation campaign. And the challenge was to identify in that data set the best antibody sequence, the variant with the best affinity compared to the parental antibody. The second challenge instead was, so as I mentioned before, the SARS-CoV-2 RBD output was also sequenced via NGS. We identified the three most abundant clusters. What do I mean? Like, we identified three clusters with the largest number of antibody for each of the three clusters. And the idea was, based on that data set for these three clusters, inside each of the three clusters to identify the best affinity antibody, the antibody with an affinity within that cluster better than the most abundant antibody in that cluster. In the first challenge, once more, based on the entire data set of the sequence selection output against RBD, the challenge was to de novo design an antibody with the best affinity, but that antibody has not to be present. The sequence of that antibody is a sequence which is not present in the dataset. So you can use the dataset to be inspired for the design of an antibody that is not present in that dataset with the best affinity possible. So here you can see different kind of organization, 29 of them applied to the challenge, and they submitted over 520 different antibodies. So, and they span from non-profit and academic institution to large biotech and pharma, AI biotech and small to mid biotech, but also big tech pharma, although very little of them. So we are wrapping up the data analysis. This competition started about 10 months ago, and we think that we are going to have a follow-up publication by the end of the year. But the take-home message that I can spoil for you was there was no AlphaFold moment. What I mean with that was there was a similar competition some years ago about identifying the best software for protein prediction, structure prediction, and AlphaFold eventually became clearly the winner of this particular competition. So we didn't find any group or any algorithm that was clearly the winner across all the 3 different challenges, but we still got some very strong data and some very strong performances. So for challenge 1, I think the starting affinity of the parental antibody was around 100 nanomolar, and one group was able to identify an antibody with 95 picomolar, so a variant of the parental antibody with a very significant improvement in terms of affinity. For the challenge number 2, so those are 27F, 28F, those represent the 3 different clusters. And so among these clusters, the competitors were able to identify some remarkable affinities. For challenge number 3, the de novo synthesis, where there was also some good performance, where we're able to de novo design an antibody with a remarkable affinity. So we are very eager to try the latest iteration of PacBio, their Kinnex platform. So as I mentioned very early during my presentation, I mentioned and I described the sequence between MySeq and Illumina and what we call standard PacBio. What Kinnex is promising, and we are eager to test it, once, of course, retaining pairing sequencing, so VL pair with the corresponding VH. What we are really eager to test is there's going to be way deeper reads number, so 10 to 100 million compared to the 2 to 8 million of the standard PacBio. Of course, it's going to retain the high fidelity accuracy, and the library preparation seems very streamlined and scalable, so it's automation ready if you want. And the ideal use case for us in our discovery pipeline is to further validate and QC our naive libraries, but also considering how deeply we can sequence, we can multiplex several final selection outputs by using this platform. So this is a proof of concept study that we are starting really soon with PacBio. So as I briefly mentioned before, we are going to test 2 different libraries, meaning those libraries, the diversity in the ACDR3 comes from 10 different set of donors. What we're gonna do, we are gonna challenge those 2 libraries against the same antigen, the same target, and we're gonna see, thanks to the use of Kinnex, how similar or how different are the antibody obtained from 2 different libraries challenged against the same target. Not only we're going to do that, but we're also going to compare the SmartBell system, PacBio, what I was calling the standard system, the one where we have been using until now, with the Kinnex library, the Kinnex platform. Not only that, but considering that now we can go even deeper, we want to further validate the functionality of our library, and also we're going to really want to test the uniqueness of 2 libraries, and we are expecting to be so, and we're going to do that with PacBio. So we can go deeper into the library validation, not just at the CDR3 level, but we are going to have the whole picture of the functional VL and VH pairing. And now I'm open to questions.
Hi, everyone. Welcome back. So yeah, we have time for questions. if you have questions, please submit them in the question module on your interface. Maybe I have a question that's very general, maybe, Fortunato. So what happens, it's very basic, right? But what happens after you identify a handful of good clones or antibodies? Can you talk about next steps in the development process for the attendees here today who are maybe less familiar with the process?
Yeah, sure. So the beauty of the possibility to obtain such a large data set of antibody after a selection campaign and the progress made in gene synthesis and recombinant antibody expression and purification allows us to go from identify the antibody sequence to clones that we can further validate in about 3 weeks. So then it depends what kind of project, but usually for potential therapeutic leads, we will move ahead with some functional assay, meaning measuring affinity and also the developability profile, aggregation, basically how those antibody behaves before going even further deeper into animal study or preclinical models, if you want.
Actually, that brings me to the next question. So what are these antibodies used for? What are the key applications? Would you say they're used as drugs or for diagnostics?
Yeah, so the vast majority of our clients, they are looking for potential therapeutic antibodies. So they have a target in mind and they're looking for antibody with specific characteristics, meaning affinities, good developability profile. So with developability, what I mean is like how an antibody behaves when it has to be manufactured, if it aggregates a higher concentration, which are usually the concentration that people use for therapeutic purposes. But we also have done, of course, selection strategy campaign where the goal is to have antibody to be used as a reagent, if you want, meaning diagnostic reagent, but also for pharmacokinetics studies. Like sometimes we develop a therapeutic antibody, and in parallel we also develop antibody that can help study the efficacy of this particular antibody in preclinical studies.
Oh, interesting. You talked about the lack of correlation between the abundance of the clones and the affinity. And so here's a question from the audience. How do you differentiate the true rare clones from any bias that might be introduced from sequencing or PCR?
Yeah, that's such a good question. So basically, first of all, our quality, the famous Q value is relatively high. So before making a call for any nucleotide, and of course the amino acid sequence generated after the nucleotide sequencing. So our threshold is relatively high. And then also we are collecting data. Also the abundance in a selection campaign allows you to understand if a clones can be considered worth to explore or not. And also depends by a selection campaign, meaning some antigen, they drive the population that we obtain after sequencing is highly dominated by a few clones. And so that's also, sometimes we increase the threshold of confidence because most of the time when a few clones are dominant in our selection campaign, the ones that might be worth to explore with a more spread population, it might be more like artifact in a population that is dominated by a few clones.
Got it. And the next question from the audience here kind of fits with what you're talking about. So what's the diversity that you can cover with one SMRT Cell, meaning the number of unique sequences, not total number of sequences, so unique sequences?
Yes, so that's of course it depends also about what kind of selection campaign we're performing, meaning like some selection campaigns, since we're looking for very specific functionality of the antibody, maybe competition with another antibody or targeting a specific epitope, the diversity is relatively limited. So we're talking about in the 10 to 50 antibody for a particular campaign. When it's more a broad campaign, like for instance, looking for antibody recognizing RBD, and where it's really diversity that we are considering, usually we can see between or even up to 1,000 different clonotype, different antibodies in our selection campaigns.
Over 1,000, wow. And then there's another question. So in the NGS data, how can you segregate between binders that are specific to the target and the non-specific one? They want to know
Yes.
that we always need a Sanger for that, Sanger results for that.
No, basically the way we work by combining phage and yeast. So we are pretty sure that our population that we submit for sequencing is composed most of the binding antibodies. That's of course based on the results that we see when we perform at the population level, thanks to yeast display and flow cytometry. That doesn't mean that all the antibody that we identify for sequencing are considered might be binders, because there might be some attrition where we go from an antibody display on the surface on yeast when we make as a full length IgG. But usually our attrition rate is usually 90% of the antibody that we call binders, they reveal to be binders when we move into reformatting them as IgG.
Got it. So thanks for everyone for staying a little bit over the time. I also, here's another. So do you take special measures to avoid a library unbalance, which can reduce the diversity that you can cover with one SMRT Cell? This question pertains to the analysis of a non-selected library.
Yes, so what we do, basically, we have these individual sub-libraries that compose our library, and every time, both when we generate positive data set in terms of target and negative data set in order to identify real negative non-binders, we keep all those libraries separated. So we try not to mix the library. Of course, there is also inside the library, there might be some biological bias for some clones that overtake the population, but we have never seen this kind of behavior. We think that it's also due to how well engineered is our library. So all the clones have the same chances to be well expressed as in phage at the beginning of the campaign and in yeast towards the end, when we are fine-tuning the binding property of those antibodies.
Got it. I think the AI antibody challenge is so exciting. I think maybe it was a little bit disappointing that there was no AlphaFold moment. But I wonder, do you think that in the future AI will be able to predict antibody binding?
Well, everybody is putting a lot of effort, including experimentally, but also money-wise. As I mentioned in the presentation, some of our clients are some of the leading AI company, and they want good quality data. So right now, I'm also biased because I do experimental work, but I think AI can really help in the short term. After we do a selection, we obtain all this data in sequencing to identify the antibody with best properties in terms of developability profile, and is somewhat also epitope recognition. De novo synthesis based on just AI approaches. The feedback that we have, both with the competition, but also with talking with potential and actual clients in the space, is like they really need more and more good quality data to train their algorithm. So probably we are going to that direction, hopefully after I retire.
Okay. Well, with this, I would like to thank you. We're a little bit over our time. So thank you, Dr. Ferrara, for your time today and sharing your experience with us. Just as a reminder for all the attendees of the webinar, following this, you'll receive a brief questionnaire. Please do take the time to fill it out. So thanks again for joining us today, and I hope you'll join us for a future webinar. And with that, take care and have a great day. Goodbye.
Bye. Bye-bye.